Hello, good afternoon to everybody. Welcome to this Friday seminar. So it's a great pleasure to introduce Eduardo Rocha today as the speaker. Eduardo did his PhD in the University of Versailles, and shortly afterward, in 2000, he became a junior researcher in the University of Paris and the Institut Pasteur, where he has stayed and is now head of the microbial evolutionary genomics research group and head of the genomes and genetics department. And throughout his research, he has contributed extensively to the understanding of bacterial genomics with several interests. He has focused mainly on bioinformatics approaches and is interested in bacterial genome organization, mobile elements, including interaction of bacteria with phages, genome dynamics, and genomic basis of pathogenesis, and probably more also. And throughout this research, he also contributed and developed with many tools to analyze bacterial genomes. And today is going to talk about horizontal gene transfer and mobile elements in bacteria. So Eduardo is here today not only giving the IGC Friday seminar, this is also is also a SymbNET online seminar, and that is because Eduardo has kindly been part of the advisory board of SymbNET. And also today, Eduardo will participate in the host microbe interactions PhD course at IGC. So thank you very much for accepting all of these invitations and for the seminar today. And I'm looking forward to it.
Thank you, Rich. Thank you very much for the invitation. Good afternoon for everyone. And so my talk today will be on the mobile elements, how they interact antagonistically, sometimes symbiotically with bacterial genomes, and how that shapes bacterial adaptation. So during my talk, I will present you a few key actors in this interplay, and the first one is the bacterium, and I call it the fickle bacterium because the bacteria that we are most interested in are these bacteria that quickly oscillate from an evolutionary point of view between bacteria that are environmental or commensals or even mutualists, and sometimes they evolve to become pathogens. And a very classic example of that is E. coli, which is probably one of the best known organisms on Earth. That can oscillate very quickly between commensalism and pathogenicity. So if you compute the infectious dose at 50% of E. coli, this is how many cells you need to uptake of E. coli to become sick with 50% probability once you're a healthy human individual. Some lineages of E. coli can make you sick with 50% probability if you just uptake 10 to 100 cells. So they're extremely efficient at producing an infection, whereas other strains of E. coli are much less efficient and they have infectious dose of 10 to the 8, 10 to the 10. And most E. coli strains actually will not make you sick if you're an healthy human individual. And this is the case of E. coli K12, which is the best studied strain. And this is an important evolutionary and medical problem because all of us live with about 1 trillion E. coli cells in our gut. And so it's really an interesting problem to understand why and how these bacteria can oscillate between being a commensal and being a deadly pathogen. Now, the reason why this occurs and this happens so frequently is that bacteria are extremely diverse, and I can represent this with a paper we just published, where we sampled about 1,300 natural isolates of E. coli all over Australia, different environments. There are a few clinical isolates, but most of them are not clinical isolates. They're different animals, including humans or the environment. And once we sequence all these genomes, one realizes that the average E. coli genome encodes about 4,600 genes. But beyond this average genome, there's only about 2,200 genes, which are what we call the core genome, and they're present in at least 99% of the strains of E. coli. In contrast, there is a pan-genome that is a diversity of different gene families that is huge. In our data set, it's 76,000 different gene families, which is 15 times larger than the average size of an E. coli genome. So in a sense, even though E. coli is very often taken as the model for other bacteria and for certain mechanisms, the model for living beings, and no E. coli strain is really a very good model for E. coli because they are so diverse, they have very different gene repertoires. So when you take these 2,200 genes of the core genome and you align and you make a phylogenetic tree, you get something that is fairly robust because there is a sort of a statistical average in terms that allows us to trace more or less correctly the lineage, the evolutionary lineage of the species. But beyond this average, there is actually a huge diversity in terms of gene repertoires. And this is what I'm representing in my graph on the right. So what I'm representing in the x-axis is a patristic distance. So if you take the phylogenetic tree of E. coli, it's the distance that goes to go from one taxon to the other. So it's the length of the branches you have to go into the tree to go from one of the taxa to the other. Some strains are close, some strains are very distant. This is what I'm representing here in the x-axis. What I'm representing in the y-axis is the gene repertoire relatedness. So this is simply the number of orthologs between two strains of the species divided by the number of genes of the smallest genome. So if the two genomes have the same families of genes, this is 1 or 100%. There's no gene in common, which of course does not occur to be 0. If one genome is a subset of the other, this is still 100%. So what we observe here is the fit of this average trend. So we have more than 1,000 genomes, so we have more than 1 million comparisons. And what I'm representing is shades of concentration of points in my graph. And what we did first was to just use a spline to understand the average tendency that we observe between bacteria that are from the same sequence types. So these are very, very closely related bacteria that one usually uses the sequence types to understand the epidemiology of certain lineages of E. coli, for instance, in hospitals. What you observe when you look at these bacteria, which are very closely related, is that very quickly the gene repertoire relatedness falls from about 100% from bacteria that have diverged yesterday at breakfast to a relatively small value of about 90%. That is, even bacteria that have diverged sometimes a few years ago already have 5 or 10% of the genes that have no ortholog in the other genome. After this initial diversification, which is mostly driven by the uptake and loss of mobile genetic elements, then there is a more stable trend through time. As time goes by, when you compare more and more distantly related bacteria, you will see a decrease in gene repertoire relatedness that is more or less linear from about 90% to 80%. But what's more interesting in this graph is not so much the average trend, but the variance in this data. Because you can easily find two strains of E. coli that have diverged very early in the history of the species, that have only about 90% of genes in common, whereas some strains which are very, very closely related, they have less than 80% of genes in common. So this varies extremely quickly. This is within one species. If you compare humans and chimps, you'd have more than 99% of genes in common. So bacteria can indeed evolve very fast, and this is largely because of mobile genetic elements. In E. coli, which is not naturally transformable bacteria, as far as you can tell, these mobile elements take a significant part of the genome. And this is what I'm representing in this very simple histogram. In gray, this is the size of the genomes we have analyzed in terms of number of genes encoding for proteins. And you see that within the species, we have genomes that encode about 3,800 proteins and genomes that encode almost 6,000. So this is already a huge gap, a huge difference in terms of gene repertoire between strains. Now, if I plot exactly the same thing in red, where I just remove from my data set the plasmids and the phages, and there are many other mobile genetic elements that I simply cannot identify, especially in draft genomes, what I get is the red histogram. And you see that the differences in the averages of the peaks between the two histograms tells us that these mobile elements, phages and plasmids, occupy about 10% of the genome. But what is more interesting is that the range of sizes, here you see that it is divided by 2 once you remove the phages and plasmids. So not only these elements take a lot of space in the genome, but actually they are the major contributors to the diversification of the species, and to make that closely related bacteria can be extremely different in terms of the genome than code for. So this brings me to my second actor in my story, which are phages. And phages are basically bacterial viruses. And if bacteria are the most abundant organisms on Earth, there are typically more phages than bacteria in most environments. So there are really a lot of them. And their impact can be huge. Some Back of the envelope calculation suggests that phages kill between 5 and 50% of bacteria per day in certain environments. And if this is true for overall, for the different environments, then some back of the envelope calculation suggests that phages are responsible for the turnover of 10% of the planet biomass on a daily basis. So their impact on ecology is huge. Their impact in microbial genomes is also very important because temperate phages, those that can integrate the bacterial genome, often carry traits that can be adaptive for the bacteria. And prophages are more than a third of E. coli pan-genome, and I showed in the previous slide that in each individual genome of E. coli, you will find different phages, sometimes up to 20 different phages in the same genome. When the phage integrates the bacteria, in a sense, it stops being a parasite and it becomes sort of a symbiont, because the traits it expresses, if they're adapted for the bacteria, they're actually adapted for the phage itself and for its reproduction. Phages are also drivers of horizontal gene transfer. When the viral particles are packaged with phage DNA, there may be mistakes, and the phage may actually package bacterial DNA and therefore shuttle bacterial genes from one cell into the other. The other key actor of horizontal gene transfer in E. coli and in many bacteria are the conjugative elements, which plasmids are the most famous ones. And these are elements that are also able to autonomously move from one cell into the other because they produce a type 4 secretion system at the cell envelope that allows to transfer single-stranded DNA from one cell into the other. And at the end of the process, there are two plasmids, one in the donor cell and another in the recipient cell. Now, plasmids can be extremely variable in size and extremely plastic. And what I'm representing here is a graph we did a couple of years ago, but still remains quite valid today, that gives you the distribution of size of plasmids. And see that smallest plasmids are about 1 kb, so very small, 1 gene at best, but the largest plasmid can be more than 2.5 megabases, which means they can encode more than 2,000 genes. And this is actually more than some complete bacterial genomes. So in one single event of transfer, the acquisition of a plasmid can lead to the acquisition of hundreds of new genes and radically different novel traits. The large conjugative plasmids tend to be conjugative, so they're able to transfer autonomously between cells. The smaller plasmids tend to be mobilizable, and these are sort of parasites of plasmids. That is, in the same way that the plasmid exploits the bacteria to reproduce between cells, the mobilizable plasmids actually exploit the conjugative elements and hijack this type 4 secretion system to move between one cell and the other. And there are about 50% of the plasmids that epidemiologically seem to move around in the bacterial communities, but we don't actually know exactly how they move around, what are the mechanisms. So plasmids can be huge, they can gain and lose genes very rapidly, and they are key drivers of antibiotic resistance, of variations in virulence, and variations in symbiosis. So these are our three major actors, the bacteria, the phages, and the plasmids. And in the lab, we've been working now for about a decade on horizontal gene transfer. And we've been very much focusing on this frame of mind where bacterial genomes are adapting because they acquire genes by horizontal gene transfer. These genes can eventually mutate, rearrange. The genes that are already in the genome, of course, they can also mutate and rearrange. And in the long term, some genes will be lost by drift or simply because they are deleterious, but some will be kept. And those that are kept are the ones actually driving a lot of the bacterial adaptation to new environments and allow them to take advantage of new ecological opportunities. But there are different mobile elements and they have different characteristics. The ecological interactions they establish with the host are very different. The amount of DNA they can transfer is very different. And so we've been trying to understand if different types of mobile genetic elements are going to bring into the bacteria different types of traits, or going to bring into the bacteria traits in different periods of time. And to tackle that, we have recently studied the genomes of Klebsiella pneumonia. So this is an enterobacteria, not very far off E. coli. I mean, from the point of view of bacterial evolution, maybe certainly more than 100 million years of divergence, but still an enterobacteria. And Klebsiella is known for encoding a capsule that is very thick. And you can see this in my photo here on the left. You can see the cells of Klebsiella and this exclusion area around the cell. And this is the bacterial capsule. It's a thick layer of polysaccharides, but it's the first point of contact between the cell and their environment. And can actually become a very important barrier to the transfer of mobile genetic elements because they have to accede to the cell envelope to be eventually able to transfer the DNA into the cell. So our analysis started from about 4,000 complete genomes of Klebsiella pneumonia, and they were presented here in a phylogenetic tree obtained by maximum likelihood. The tree in itself, it's not very important. The important thing is these colors that you see around the tree, and these colors represent the predicted serotypes of the bacteria. And the serotypes are types of capsules that we identify in Klebsiella. They correspond to different sugars that, for instance, in the capsule, and they have different characteristics. And what you can see is that there are a lot, there are almost 100 different serotypes in our data set, and they're not at all monophyletic. So you see the same colors appear in different parts of the tree, and what this means is that the capsule is actually very frequently transferred and possibly lost and regained in different lineages. So on one hand, the capsule is expected to shape gene flow because it affects the ability of the mobile elements to infect the bacteria. And on the other hand, the capsule itself changes by horizontal gene transfer. It depends on gene flow to evolve. So this was a very interesting case to study what is the interplay between the mobile elements and the capsule. And the first question we put forward was, is the capsule actually shaping gene flow in such a way that bacteria that have the same serotypes, the same types of sugar at the capsule, will exchange more DNA than bacteria that have different serotypes? So this is the analysis I'm showing you here. We take our very large phylogenetic tree with 4,000 strains, and we identify recent co-gains, that is, genes that have been acquired in the terminal branches of our phylogenetic tree, and that were acquired multiple times independently in the tree. And then the question we put forward is, well, when you have these cases that genes that have been recently acquired independently in different parts of the tree, do these multiple acquisitions tend to occur in bacteria that have the same serotype or in bacteria that have different serotypes? And the answer is here. We do simulations of phylogenetic trees, and each time we simulate gains and losses, and we can compare that with what we really observe. And these are the distributions, and what they show is a very strong bias towards gene flow within the same serotype. That is, bacteria that have the same type of envelope will actually exchange more DNA ones with the others than bacteria that have different serotypes. When we exclude from our data set the plasmids, what we observe is that the bias increases. And what this tells us is that conjugation is probably not the cause of this bias. When you exclude prophages, which are one of the mechanisms that phages can use to transfer between bacteria, transfer DNA between bacteria, well, you see that the bias slightly decreases and somehow suggests that phages may be the culprit here. And it is actually known that phages have to interact closely with the capsule, and they tend to be capsule serotype specific, and they will only infect bacteria with a small number of serotypes, or even within one single type of serotypes. So I won't have the time to go into the details, but we went to the lab, and we did show that phages do tend to in Klebsiella, tend to infect bacteria of one single or a few serotypes. And actually, if you do experimental evolution, and this is published, you can go and look at the paper, and you challenge Klebsiella populations with phages, the best way of them to be protected from phages is simply to lose the capsule. Once the bacteria loses the capsule, it is no longer infected by the phages because they have co-evolved with Klebsiella for a long period of time to expect the capsule, and therefore the absorption of the phage to the cell envelope depends on the presence of the capsule. So this means that we have a sort of a gene flow driven by phages within the species that is peculiar. You have lots of gene flow between individuals of the same serotype, here as 1 or here as 2. You have less gene flow between different serotypes and very little when the strains have lost the capsule. And these strains that have lost the capsule, of course, they lost a trait that can be important, but on the other hand, they're protected from phages. Now, if they're protected from phages, one might think then bacteria would then be sexually isolated, because phages are one of the two major mechanisms of transfer or gene transfer in Klebsiella, which are not naturally transformable. And so they depend either on phages or on conjugative elements. So this we can test too bioinformatically. We take our very large tree and we look for cases like this. That is, we look for cases where one strain has very recently lost the ability to produce a capsule. So you can easily derive this by looking at pseudogenes of essential genes for the production of the capsule in the genomes, or sometimes the locus is simply missing. And then you can compare gene flow in the terminal branches of strains that have just lost the capsule with the terminal branches of bacteria that have kept the original capsule. And what you get is not at all what we might expect. That is, there is much more horizontal gene transfer towards bacteria that have lost the capsule. Now, in the previous slide, I showed you, and there is now ample evidence demonstrating that if the bacteria lose the capsule, they are resistant to most phages. And therefore, the mechanism that remains is conjugation. Now, there was very little data in the literature about conjugation and how it affects or is affected by the presence of a capsule. So we went into the lab and we took several isolates of Klebsiella. We knocked out the capsule, and then we made experiments of conjugation where the wild type or the non-capsulated variants are challenged with the conjugative plasmid encoding antibiotic resistance gene. And what you can observe is that in every single case, there is an increase in conjugation rates in bacteria that have actually lost the capsule. So now we have two processes of horizontal gene transfer acting on the bacteria, and they react very differently in relation to the presence or absence of a capsule. In terms of conjugation, what we observe is certain gene flow within serotypes, here S1 and S2, certain gene flow between serotypes, and a lot of gene flow in bacteria that have lost the capsule. These bacteria don't have anymore this thick layer of polysaccharides around the cell envelope, and this presumably facilitates the work of the conjugation pillars that have to transfer single-stranded DNA from one cell into the other. So overall, it does matter which type of mobile element it's transferring the genes. In phages, you have lots of gene flow within serotypes, little gene flow between serotypes, and almost none in bacteria that lack a capsule. Whereas in conjugation, it's almost the opposite. You have lots of gene flow towards bacteria that lack a capsule. And in a sense, this starts to explain the dynamics we observe in natural populations, where bacteria sometimes lose the capsule, and this may occur by many reasons. One of them is phage predation, as I told you. If the bacteria is challenged with phages, it tends to lose the capsule and then be protected from phages. And then there is a transition in terms of gene flow. These bacteria now, they are very receptive to conjugation and not at all receptive by gene flow associated with phages. And because conjugation allows the transfer of a huge amount of genetic information, it can lead to the transfer of the capsule locus, which is very large in Klebsiella, and therefore lead to a transition of the bacteria that was non-capsulated, and it can now be capsulated again. And either it acquires the same serotype and then it goes back here, or it gets a new serotype and then it goes here. And now it has a new serotype. It means it has enough, again, lots of gene flow associated with phages, but these phages will be different phages because this is a different serotype. And so by changing its serotype, the bacteria is changing its basin of attraction in terms of gene flow. That is, the neighbors, the individuals in the species with which it's sharing DNA changes from the ones who had the serotype 1, from now those who have the serotype 2. So there is a very complex interplay between the cell envelope and the mobile genetic elements, which is not the same for the different mobile elements. And this will affect the types of genes that are acquired and the rate at which they are acquired, depending on the physiological conditions of the cell. So the types of MGE matter, but occasionally some mobile genetic elements do infect the bacteria. And then there is a trade-off between adaptation and defense, because these mobile elements, they can bring antibiotic resistance genes, they can bring virulence factors, they can allow the bacteria to become symbiotic, for instance, with plants in rhizobia, but they also can be costly. And so the bacteria have developed ways to tackle this and to moderate the effect of mobile genetic elements. So what I'm representing in this slide, it's a picture of an E. coli cell that has simply exploded after a lysis event, and you can still see a few phage particles in the picture. And this reminds us that these mobile genetic elements that can bring adaptive traits can also be very costly. In the case of phages, they can literally kill the bacteria. So bacteria have developed for a long time tools, immune systems, if you want, that allow them to moderate, control, or filter the mobile genetic elements that infect them. One very classical one, which is the first one that was discovered, and it is actually by far the most abundant, is restriction-modification. In this system, bacteria use epigenetic markers, typically methylation patterns, that allow it to distinguish self from non-self. If the phage that infects the bacteria is correctly methylated, it is not targeted by this immune system. By restriction-modification, it will produce a progeny. But phages that arrive, that have infected other cells, if they don't have the correct methylation patterns, they will be destroyed by the restriction-modification system. So this is an important barrier to transfer, but in a sense, it could also allow a lot of transfer of DNA between very closely related bacteria because they would have similar patterns of epigenetic modifications. So a few years ago, we tried to understand if restriction modification systems were actually a mechanism that would allow more horizontal gene transfer, or on the contrary, would somehow restrict horizontal gene transfer, in which case it could be a certain sort of cost associated with an immune system. Now, to do this, and this is work done by Pedro Oliveira, who has done a job in the lab in Genoscope, we tried to understand what was the impact of restriction modification in gene flow, both in terms of allelic recombination, so change of alleles in genes of the core genome, and gain of radically novel genes by horizontal gene transfer. And the principle is very simple. We have species. Within each species, we have several strains, and we make pan-genomes and we make phylogenetic trees. And we can actually identify restriction modification systems in every genome and identify which are the patterns it's recognizing, right? The epigenetic patterns. So we can define strains which are compatible. For instance, here the whites or the blacks, they have the same type of modifications, and strains which are incompatible from the point of view of restriction, they have different epigenetic markers. And so our analysis is very simple now. If I just wave it out with my hands, it's to try to understand if within this set of species, we find more homologous recombination, more horizontal gene transfer between bacteria that are compatible from the point of view of restriction or incompatible from the point of view of restriction. And what we observe is that systematically, there is more horizontal gene transfer and homologous recombination between bacteria that have the same type of compatible restriction modification systems. This is twice as much for horizontal gene transfer and 8 as much for homologous recombination, and the results are quite constant across the species. So indeed, the presence of these restriction modification systems carves preferential routes of gene transfer, meaning that strains that have similar restriction modification systems will share more genes than the bacteria that have different restriction modification systems. Now, the problem is that it is not much of use to have the same restriction modification system for a very long period of time, because in the same way as bacteria evolve defenses, phages and plasmids also evolve counter defenses. And so it turns out that restriction modification systems keep changing in bacterial populations. And this has an interesting consequence from the evolutionary point of view. So let's take an ancestral population here in mauve, and they have one restriction modification system. They make a population, and within this population, there's lots of gene flow because they all have the same restriction modification system. DNA can flow freely between cells because it's correctly epigenetically marked. As time goes by, the population diverges, and different individuals will get different types of restriction modification systems. And as this occurs, what you observe is the fragmentation of the population in terms of gene flow. Individuals with the same types of restriction modification systems will still share lots of genes, but there will be much less gene flow between populations that have different restriction modification systems. So on one hand, there is an adaptive need of diversifying defense systems because the mobile elements are also evolving and evolving counter defenses. On the other hand, these are going to have an impact that is important in terms of gene flow in natural populations. And indeed, phages and plasmids have evolved a long time mechanisms that allow them to counteract the effect of restriction modification systems. So one that is important that we'll meet in a couple of slides ago is anti-restriction, and the most famous example is one of HT7. When the cell that is infecting has a restriction modification system, HT7 actually produces a protein which is called OCR that inactivates the function of restriction. So it's a counter defense. As the bacteria has evolved defenses, the phage has evolved counter defenses. And this means that actually the restriction barrier to gene flow is important, but it's not strict. That is, there is still horizontal gene transfer, even between bacteria that have different restriction modification systems, because mobile elements have found a way to go through. Now, for a long time, we have worked on defense systems, and it has been known for decades now that homologous systems that are defense systems in bacteria sometimes have homologs in phages and in plasmids. For a long time, this was taken as sort of a curiosity, but the results in the last 12 months or 2 years have shown us that it is much more than that, and they're somehow radically changing the perspective we have on bacterial evolution and on the role of these defense systems in shaping bacterial adaptation. And to go on, I have to present the fourth actor of my story, which is the phage satellite. So phage satellites are not actually phages, they're mobile genetic elements that are parasites of phages. So in a sense, they do to phages what phages do to bacteria, they're parasites, and they're going to take advantage of things that are produced by phages, notably the viral particles, and they're going to use it to move around between bacteria. So I'm going to talk about the P4 satellite, which is the best known and the most first one discovered actually a long time ago. And P4 is a parasite of the phage P2. When phage P2 infects E. coli, the phage P4 is integrated in the chromosome, will eventually be induced, and as the phage starts the lytic cycle and produces the concatenates of its genome to be packaged in the viral particle, phage P2, the satellite P4, produces a machinery that allows to pick the viral particles of the P2 phage and shrink the capsid such that it can only package P4 genomes. So in a sense, the P4 genome, it's small, it does not encode any structural component of the viral particle. It needs other viral particles to move around, and it does this by hijacking the viral particles of P2. So it's a parasite, and at the end of the process, when there is cell lysis, there is a mixture of virions, the traditional P2-like virions with phage DNA, and P2-modified viral particles, which have inside not the P2 genome, but the P4 genome. So parasites of parasites. And for a long time, we knew very few of these P4 elements. There were about 3 that were described in the literature. And George Souza, which is actually a former IGC member in the lab, took up this topic and realized that these P4-like satellites are extremely abundant. So if you take E. coli, 44% of the genomes of E. coli encode at least 1 P4 element, and some can encode up to 3. So these are extremely abundant elements. We have now identified more than 1,000, where we knew only about 3 last year. And then now that we have 1,000, we can do to these P4-like elements the same that we did at the beginning to E. coli. Is we can put them all together and identify core genome and the pan-genome. And this is what you get here in my figure. This is the core genome of the satellite P4. It has 7 genes, and the genes are very conserved in terms of gene order. And this is what you see in my graph. The edges in my graph are very thick if the 2 genes are contiguous in the backbone of P4. Now, there is also an extensive pan-genome for P4 elements, even though they're quite small. And you see that it's not uniformly distributed along the genome. This is the pan-genome that we have observed. And in this part of the P4 element, there's a huge diversity of gene repertoires. We found more than 200 different gene families in this region. Now, in each individual element, there are only 2 or 3 genes at most, but different elements have radically different genes at this precise location. And it turns out that most of these genes are actually what one would call bacterial defense systems. So we took all these genes and we made sort of a taxonomy of the functions we identified there. And about 3/4 of these genes are known or putative defense systems in E. coli. So this is the taxonomy of these elements. We find some that are very well known, like restriction-modification systems here and there and in other places. But many of them are putative, but the protein domains we observe are compatible with what we know of bacterial defense systems. So our collaborator, David Bicard, picked a few of them that were not known, cloned them into an E. coli cell and challenged them with a number of phages. And this is what you get. you observe that in all the cases, these elements do provide defense against one, two, or sometimes many different phages. So these P4 phage satellites, which are parasites of phages, are actually protecting the cell from other phages. Now, one of the elements that caught our attention is the Perry system here, and it provides resistance. These are the systems here. There are a few variants, and provides resistance against a number of phages, and it was not at all known in the literature. And so we tried to inquire on its function. So there is a preprint in bioRxiv. If you want to go into the details, I'll be very brief today. So we went to the lab, and this is the growth of E. coli carrying in red and not carrying the Perry system. And in the absence of phages, you have the growth curves here. And you see that the strains that don't carry the Perry system tend to grow slightly better. And this reminds us that immune systems are costly, right? These are elements that are probably expressed constitutively, and they have a cost. But they also provide rewards. If you challenge the culture with T7 phage, you can see it here. The population that has the Perry system stabilizes. It is somehow protected from the phage, whereas the bacteria that lack the Perry system actually collapse and they disappear. They are wiped out by the T7 element. So some further work showed us that actually the Perry system is an anti-restriction system. So E. coli produces restriction-modification systems, T7 produces anti-restriction systems, which are, in this case, the OCR protein, and some E. coli produce the Perry system, which is a second layer of defense that will actually induce cell death or at least cell growth arrest if the first layer of defense has failed. And it's a second, more radical mechanism that the bacteria will use to tackle the infection by phages. It might lead to cell death, but will prevent the epidemic of the phages to propagate in the population. Now, the thing is that this is a bacterial defense system, that it's not very bacterial in the sense that it's encoded in a mobile genetic element. And there is just one region in the phage P4 that is in the satellite P4 that is homologous to the phage P2, the phagid parasites, which is the cos site. And so we went to look in the cos site of the phage P2 to see if there too we could find defense systems, that is systems that we typically call bacterial defense systems. And indeed, in about 2/3 of the cases, in this exact location in the phage genome, we find systems like restriction-modification systems and other systems that are known to be defense systems. Some are new. David tested a few of them in an E. coli strain, and they do provide resistance against other phages. So now the situation is fairly complex. We have in 2,500 genomes of Enterobacteria, more than 1,000 P4-like elements, more than 2,000 P2-like prophages, and all these elements are actually in relation, in antagonistic relation, either with the host, which is for the P2 phages is the bacteria, or in the case of the P4 is P2 phages. So lots of opportunities of antagonistic interactions, and there may also be some mutualistic interactions in the sense that it may be that very often the interests of P4 and the cell are aligned, and that the interests of P2 and the bacteria are aligned. And this is possibly why these elements are so good at protecting from other phages, that by killing the cell, we'll also kill the host of P2 and P4. Now, in the last couple of months, a few papers have come out showing the same thing that we've been showing in P4. And of particular interest is a paper recently on SXT, which is an element, it's a mobile genetic element, it's a conjugative element. And I'm representing here the picture they showed in the paper. And in one location, a hotspot in the mobile genetic elements, they also found a bunch of bacterial defense systems. Martin Poles recently published in Science that in vibriophages there are many defense systems. Jane Eaton recently published in Salmonella prophages. There is also phage defenses, and we also have a bioRxiv that will hopefully very soon come out, showing that in Vibrio Australia, about 72% at least of the defense systems that we would call bacterial defense systems are actually encoded in mobile genetic elements like phages and plasmids. So we come to the conclusion that most of the bacterial defense systems are actually not bacterial at all. They are encoded by mobile genetic elements that move around and that occasionally defend the bacteria because that's in the best interest. And this actually radically changes the way we look at these defense systems and the way we can understand this interplay, these ecological interactions between the bacteria and the mobile genetic elements. We started in a very simplistic way. We have a host, we have a parasite, and then we have defenses, we have counter defenses, we have counter defenses, and we know at least of one case of a counter defense. And I'm sure there are more. But now the situation is much more complex and in a sense much more interesting, because what we have is that in the bacterial genome are coexisting lots of different genetic agents. They can be phages, they can be conjugative plasmids, they can be parasites of conjugative plasmids like the mobilizable plasmids, they can be parasites of temperate phages like the satellites, and all these elements are interacting. Sometimes they have common interests. It is in the interest of most of these mobile genetic elements to protect the cell from virulent phages, because they will kill the cell, and by killing the cell, will also kill these mobile genetic elements. But of course, the many other interests of these agents are very different. Some are parasites of the others, some are parasitized by the others. And this leads to a very complex network of interactions. That means that when we observe a defense system of the bacteria, it is not at all clear who is defending. Is it the bacteria? Is it the mobile genetic element? And it is not always clear who's being targeted. Is it the virulent phage that we typically use to test the function of these defense systems, or is it something else, or is it an ensemble, a set of different mobile elements that are important and antagonizing the mobile genetic element that is encoding the immune system? So to finish, I started with somewhat simplistic view of the bacteria that adapts because there is lots of horizontal gene transfer, which may eventually be modified by rearrangements and mutations. And a lot of this DNA is eventually lost because it is not adaptive. And we move on to a situation which is much more complex, where we have a sort of a cosmos within the cell of very different genetic agents that have their own evolutionary interests. They can ally themselves for certain, because when they have common interests, they can also antagonize themselves. Some are parasites of the others. And this makes the situation much more interesting from the point of view of evolutionary biologists, because it raises key questions that we thought, for some of them at least, were solved, and actually they're not solved at all. And some of them are listed here. Some of that are interested us the most. What is the interplay between the cell physiological traits, notably the cell envelope, these mobile genetic elements, and the acquisition of traits like antibiotic resistance? And this can be fairly complex. Bacteria with capsules are more resistant to antibiotics than bacteria lacking capsules. But then the bacteria that lack capsules may more easily acquire conjugative elements, and antibiotic resistance genes tend to be encoded in conjugative elements. So it may be that the bacteria that lack capsules are more susceptible to antibiotics initially, but they are faster at getting antibiotic resistance genes. So this really changes the perspective we have on things like who's defending whom from what, as I mentioned before, or which types of mobile genetic elements are transferring specific traits. It is well known in the literature that conjugative elements tend to transfer antibiotic resistance genes, whereas phages very rarely do so. And why is that? We don't really know, and it's probably in relation with the ecological interaction of the phages and the bacteria with the genetic capacitance of the different mobile genetic elements, and possibly also with their genetic plasticity. And finally, it is very tempting now to do for bacteria and their mobile genetic elements what we do for humans and viruses like COVID, or for bacteria like E. coli, and try to use the concepts we have from classical epidemiology theory to try to understand how these elements propagate in communities and eventually spread adaptive traits, or from an inverse point of view, they can just decimate bacterial populations by infecting and killing them. So these are ongoing questions in our lab, I think in many other labs right now. So to finish, I would like to thank the people who have done this work. So this is our lab in Pasteur Institute. These are the people in the lab. And I would like especially to thank for this talk the work of Marie Touchon on comparative genomics of E. coli, the work of Jorge Souza on P4 satellites and the capsule interaction of the capsule with phages. Pedro Oliveira, who did all the work associated with distribution, or most of the work associated with restriction-modification systems in bacterial populations. Mathieu Diquet and Olaya Randuelas, who have been leading the work on the bacterial capsule in Klebsiella pneumonia and its effects on horizontal gene transfer. And our collaborators specifically for this work, David Bicard and François Rosset for the defense systems in P4 and in P2, Frédéric Leroux for the defense systems in Vibrio, and Eric Denamure and David Gordon for the work on E. coli diversity in Australia. And I'll be glad to answer to your questions. Thank you.
Thank you very much, Eduardo. Are there questions? You can raise your hand, write in the chat, or open the mic.
Hello.
Hi.
Hello, can you hear me?
Yes.
Yes, Isabel here. Very nice, Eduardo. Thanks a lot for the talk.
Thanks,
So
Isabel.
I'm wondering, when you were discussing the capsule and the phages, that if you wonder about the importance of the host immune system in driving the capsule evolution, since at the end of the day, right, they are these serotypes, and the capsules are supposed to be important for the big host small bacteria interaction, you know, as much as for the small host bacteria phage interaction, no? So is there a role for the host immune system to think about this diversity in the capsules?
There is, but I'm not quite certain that it's the host that's driving the diversification of the capsule. And this is because in many bacteria, actually, the cells that have a capsule or a thicker capsule tend to be less immunogenic than the others. And the reason is that the capsule hides lots of proteins and even the LPS that can will be recognized by the immune system. So the capsule is actually a tool that the bacteria use to escape from the immune system. Of course, it may be that the immune system somehow learns to recognize the capsule, and therefore this produces a selection for diversification of the capsule. But my guess is that the phages also have an important role in diversification of the capsule, because once the community gets filled with phages specific for a given capsule, the best way of the bacteria to get rid of these phages is simply to get rid of the capsule and then get a new one, right? So for us at this stage, it is not very clear what is driving capsule diversification, and my best guess would be that it's phages and not the immune system of the bacteria. Of humans, for instance.
Right. So if that is correct, if you would be studying some bacteria from the sea where there's no immune system, right?
Yeah.
Then you should get to the same conclusions for species if you had a large collection of bacterial species from the sea, no?
Yeah, we should. But the problem is that serotyping is something that has been developed mostly for pathogenic bacteria because it was one way actually of classing them for a long time. So it's not so certain, but what we do know is that commensals like Bacteroides in the gut have capsules, and they have inbuilt mechanisms to diversify the capsule. And there is not so much evidence that it's because of the immune system, but we do know they're targeted by phages. So I think the jury is still out, but I would really tend to think that the capsule is diversifying for something else. And we do know that actually the capsule is more frequent in the environmental bacteria than in pathogenic bacteria.
Okay. And
it's actually quite rare in obligatory pathogens. So
that's a point, yeah. So the other question, if I'm allowed, is related with the defense, the last part of your talk about the defense. So you find out this huge diversity of these things that are defense systems, or part of them. So why is it the case that, you know, what would be the optimal number of defense systems for a bacteria? They can move around, as you showed, right? So do the phages, could we come up with an optimal number of defense systems that an optimal bacteria could have?
I don't know. So that's a very interesting question. The thing is that it is not just about the bacteria and the phage, right? And the thing is that this is about bacteria and the mobile elements and the phage. And so the number of the defense systems that you'll find in the bacteria is just not the optimal number that is associated with the sort of immune defense of the bacteria, because actually what we're observing here is defense systems that are from the mobile genetic elements, and so they are selected by the mobile genetic elements. So it may even be that some of these are actually very deleterious for the bacteria. When you have a phage that will kill a bacterium, for instance, there is an infection by a satellite, if that occurs, it's actually very deleterious for the bacteria. If you have plasmids that kill the bacteria as a means of defense, because another plasmid has infected the cell, this is can be extremely deleterious for the bacteria. So I'm not sure that you can use these kinds of criteria of optimality to know if there is a perfect number of defense systems in bacteria. I actually suspect we cannot, right? There might be one if this was just really a bacterial thing. But since this is mostly not a bacterial thing, because most of these defense systems, at least in certain species, seem to be in mobile genetic elements, I don't think we can really say that.
All right, okay. So one last, if I can. So given what you've found out now, specifically in E. coli, what would be the strain that you would recommend to study in the laboratory that is, good enough to try and do some kind of experimental evolution on these nice antagonistic interactions?
There is no good strain, right? So now in the work that we've been starting in satellite and so on, we've been thinking of it, and I think we're going to go for very simple vector strains, right? The simplest it is the best, because if you take some E. coli strains can have up to 18 prophages and several plasmids and many genetic elements integrated, and this is just a hell of a complication in terms of the possible defense systems, right? So I would even say there is this E. coli strain that has been stripped from everything that's accessory. They have removed 20% of the genome and most mobile genetic elements, and maybe one should start there.
All right. Okay. Thank you, Eduardo.
Yeah.
Any more questions? Oh, there is from Nelson Frazão. You want to ask the question or should I read it? I'll read it if you don't. Okay. So Nelson Frazão is asking if the capsules impact the capacity to colonize, which
yes,
yeah,
the capsules
impact in the pneumonia, yes. So thicker capsule leads to lower ability to colonize.
Yeah, I think they impacted it a lot. I'm going to stop sharing now. No point in you can look at me. So they do. When we did an experiment, a computational analysis for that a few years ago, it was published in PLOS Genetics or PLOS Pathogens. And what we did was the following: we took a very large number of metagenome studies, and we identified the presence of bacterial species that we knew had a capsule and we knew don't have a capsule. And we tried to understand if the bacteria that have a capsule are identified in more environments or fewer environments than the bacteria that do not encode capsules. And what we observe is very clear. The bacteria that encode capsules are found in more diverse sets of environments, metagenomic data sets, than the bacteria that lack a capsule, that are much more constrained to a small number of environments. So yes, the capsule is a key colonization trait, at least in our opinion.
So can I ask something also about the capsule? So once you have a group of bacteria that have the same capsule, they are subject to the same phages. But if they have prophages integrated already, they will also be resistant to those phages. And I suppose horizontal gene transfer will still happening when phage-like particles move with neighbor genes to the phage, right?
Yep.
They won't be resistant to those integration events. So can you see this group of bacteria with the same capsule as a sexually compatible bacteria? And that's what makes the difference between the capsules is
yeah.
the cohort.
Exactly, that's exactly how we see them. The presence of similar capsules, so serotypes, or the presence of similar restriction modification systems, in a sense, it's quite similar. They will produce pairs of genomes that are more compatible than others. And therefore, they will tend to drive, to fragment the gene flow within the species, such that these pairs will exchange a lot and much less with the other bacteria. Now, the thing is that you have the capsule serotype, you have different defense systems. And so this can lead to a complex combinatorics, right? So it's not so simple that two bacteria, because they have the same capsule, will always exchange lots of genes and the others won't, because maybe that sometimes the capsule is expressed or it's lost, and there are all the other traits that are going to affect gene flow that will also determine if there is a transfer between these two individual cells. So it's not so simple, but basically it's exactly as I said. There is preferential gene flow between bacteria that have compatible structures in the involved and those that have similar defenses.
Any more questions?
Yeah, I have a question.
Yes.
Can you hear me?
Yes.
Yes.
Okay. So earlier in your presentation, you showed that capsule-free bacteria have more gene gains than those with a capsule. I guess I have a question about how you estimated the gene gain in this case, because my question is, horizontal gene transfer also has this ability to continuously revert gene loss if it's maladaptive gene loss. So if that happens, won't you maybe underestimate gene gain because you miss out on this rapid turnover mode of continuously losing and gaining genes? So there is a lot of gene gain, but you don't see it?
Yeah, sure. I mean, most of the gene gain, we don't see it because it's either neutral or deleterious, and it will disappear at right after, right? Or the lineage is dead, which is basically the same from evolutionary point of view. So we don't see most of the gene transfer, but in the case that you're mentioning, what we did was to compare terminal branches that have kept the capsule and terminal branches that have lost the capsule, right? And it so turns out that in our tree, the length of these branches of the two populations is the same on average, okay? So we can compare them directly. So this means that we are missing a lot of transfer, but we should be missing more or less the same amount of transfer in both types of branches.
Okay. Yeah, okay, that makes
sense.
So
this is why we can make the test, otherwise we couldn't.
Yeah, okay, I get it. Thank you.
Okay.
Okay, any other question? Okay, if not, thank you very much, Eduardo.
Thank you.